What I'd like to talk about this morning is water tracing experiments in the critical zone. And I want to acknowledge my co-authors, Havame Evaristo, a PhD student who's about to complete here at the University of Saskatchewan, and then Natalie Orwelski, a postdoc who's just finished and is now an assistant professor at Gießen University in Germany. Diane Pratt, a research engineer with me here at the Global Institute for Water Security. And then finally Bing Si, who's collaborated on parts of this presentation and I know is with you all today. So thanks again for having me. I also want to acknowledge collaborators on a few different parts of the talk. The Tropical Rainforest Biome at Biosphere 2, some of our China tritium work that I'll present, and then some of the lab extraction work that I'll present run by Kim Jansen and Cody Miller, both at the Institute. So what I'd like to cover today are a series of points related to hydropedology and ecohydrology. And if we look at a cartoon like this, what I want to try to tackle is this link between water that makes its way into the soil and is extracted by vegetation or continues its downward or lateral migration to groundwater and the stream. So I'll start out with some recent findings. This is work that's been published in the last 12 months or so. I'll talk then about some of the challenges for water extraction, particularly of soil water, and then some new ongoing work, fairly preliminary, from Biosphere 2 and our work in the Loess Plateau. So first then recent findings. Back in September we published a paper that was a global compendium of work that had looked at soil water, plant water, and other waters to test something we call the ecohydrological separation hypothesis. Before I show you that, I just want to give you kind of a cartoon representation of this. I touched on this, I believe, at the last or hydropedology workshop. And the idea is here on the left-hand side, global rainwater, meteoric water, falls on the meteoric water line. And stream flow, groundwater recharge, mobile soil water you might sample with a suction lysimeter or a tension lysimeter fall on this slope of the meteoric water line. And stream flow is a lagged and damped or compressed signal. Then if you look at the right-hand side, what we found in a 2010 paper led by Renee Brooks and myself and a couple of others in Nature Geosciences is that the plant water is following not the global meteoric water line, but a soil water evaporation line. And this is at a slope less than the global meteoric water line. And the plant water, like the stream, is a lagged and damp version of that source water, and this is the tightly bound soil water. The water you might get out if you did cryogenic extraction or centrifuging. This is water held up to 10, 15 megapascals. This contrasts with the mobile soil water that would fall on the meteoric water line on the left-hand side. Now, when we did this global kind of meta-analysis, if you like, here's Havame in the upper right-hand corner, what we found was, as expected, when we look at this relationship between deuterium and O18, global groundwater, global stream water is hugging that meteoric water line, the global meteoric water line. But when we look at plant water from these studies, when we look at soil water from these studies, these are plotting below the line. And again, following a slope that's shallower than the meteoric water line, consistent with this early idea of an ecohydrological separation. We quantified that offset rather than just the visual that I showed you in the previous slide, using something quite straightforward, that is looking at the slope and y-intercept of this relationship between deuterium and O18, and then including this S-term, standard deviation for measurement uncertainty. So what we found was shown on this graph, where now the y-axis is the precipitation offset. These are the different study sites that you see along the bottom, grouped into ecohydrological zones, if you like. 0 would represent falling on the meteoric water line, and you can see that the stream water and groundwater samples are largely hugging that line. But the plant water and the soil water are much more negative, falling away from that line, suggesting that they are on a quantifiably different slope, and suggesting that these are a different pool of water in terms of what's contributing to the more mobile water forming groundwater recharge and stream flow. there's still many issues with this hypothesis. It's a hypothesis. I'm actively seeking to reject it in work that I'll show you a little later on. But what's interesting, about the same time as that September paper that I just presented, there was one by Stephen Good and colleagues that came out in Science, showing effectively similar kinds of findings, but with very different methodology. This is work from the AURA satellite. This is looking at atmospheric vapor and looking at the isotopic composition of that vapor. And to close the vapor balance globally, they've invoked this two water worlds hypothesis to achieve mass balance. This is the paper, Hydrologic Connectivity Constrains Partitioning of Global Terrestrial Water Fluxes. And just to quote from the paper, they're saying, or their finding was that limited connectivity between soil and surface water fundamentally structures physical and biogeochemical interactions of water transitioning through catchments. And this has been a big issue. If water is in fact not mixing the way we might think it would in the critical zone, then this has enormous biogeochemical implications. And interestingly, just a few months ago, there was a paper on this topic, kind of invoking two kind of nitrate worlds hypothesis, as it were. This was led by Stephen Hall. This came out in Ecologia in the spring. And I'll make perhaps a quote from this paper, this very interesting work. Soil extraction sampled in the anaerobic microsites inside soil aggregates where denitrification can occur, whereas lysimeters largely reflected the transport of N that was nitrified in the aerobic macropore environment or in the surface litter. So again, identifying these two pools that are not mixing, which is really the heart of these findings since about 2010 using this dual isotope approach. But this whole business is not without its challenges. There's a whole issue of what are the physiological explanations for this behavior, plants going after perhaps water that's different to the mobile water. There are issues with extractions in terms of how that impacts interpretation. There are scale issues, given the fact that mycorrhizal fungi are operating at scales many, many orders of magnitude smaller than we typically examine when we do these kinds of extractions. And there have been a couple of recent and very useful papers published, this one by Otter in Journal of Hydrology in 2014, really alerting the community to fractionation effects associated with cation exchange. This is linked to Todd Dawson's group at UC Berkeley. And then also at UC Berkeley, work published by Jasper Ocean, one of Bill Dietrich's PhD students, along with Bill and Todd and Inez Fung, showing really interesting rock water use of vegetation at their site in California. And I'll just read you a small quote here saying, from this paper, these differences suggest that the use of water isotopes as tracers must consider the possibility of subsurface isotope evolution and the influence of exchange with more tightly held water. And Bill might have talked about this on Wednesday when he gave his talk at the meeting. What I want to do is talk not about these different pools and perhaps basic biogeochemical, isotope biogeochemical issues, but I want to go to the extraction method itself and just think critically about that for a moment. This is some of the postdoc work of Natalie Orlowski shown here. And we're doing a very simple analysis here. Look at the red dot, and I'm going to emphasize that red dot for you. So that red dot is showing you the label water. This is a lab standard water, and what we've done is we've taken a sandy soil and we've oven dried that soil, and we've ensured ourselves that we've extracted all the water from that soil. We weigh it before and after. And then we do extractions. You can see down here in the bottom right-hand corner, 120 minute, 180 minute. And what we're able to do is to convince ourselves that in fact we can get that labeled water out of the soil once we re-irrigate it. But if we do the same experiment now with soils that contained some additional amounts of silt or clay, here's what we find. And this is the same label now. We're trying to get back to that same label, but you're seeing then for these different extraction times, for ever increasingly clayey soils, if you like, going down to the bottom left-hand corner, we're not able to get back to that signal. And this is a problem, and this is a problem associated with what is our standard technique in most of this business, and that is cryogenic extraction. The idea is that you're heating, cooling, and you're able to get the water out of the soil, coax it out down to basically wilting point, or 15 megapascals or thereabouts. Now, if we look at these extraction systems, here's a photo of several. Upper right or upper left-hand corner is one that was developed at Gießen. We've been using in the lab here in Saskatoon, but there are many others, and I won't bother to name them or maybe discuss some of their attributes. But what we've done is a global intercomparison of cryogenic systems, just to see what effect the system type might have on our extracted water. So this next slide shows the results of an analysis that we're still working on. We just got these results. And you're seeing now 16 labs that use these different approaches that I showed you in the previous slide. And what we're seeing are the red dots in the two slides I'm showing you are the reference water. So they all had standard reference water. They all applied the same temperature for drying. We had extraction times, you see up in the upper left-hand corner, for two soils, a silty sand and a clayey loam. And all labs followed standard procedure. I've kind of shaded the lab names in the bottom to protect the innocent. And what you see is that on the left-hand side, these are extracted soils with a water content of about 8%. On the right-hand soil side, these were wetter soils. They were extracted at 20%. They're not all falling on that red dot. There's enormous scatter. You're seeing it from, gosh, just on the right-hand side from -12 per mil to maybe -7 per mil for O18, and for deuterium -90 to -50 or so. This is challenging because we use cryogenic extraction as effectively our standard, and not only are there issues associated with these things like cation exchange, like organic material, there are lab-to-lab variations that we need to be thinking about. We're going to now look at how these cryogenic extractions compare to perhaps other methods that are out there. So for instance, here's the cryogenic extraction, but there are new methods coming online all the time. Neil Munsgaard at the James Cook University in Cairns about a year and a half ago came out with a microwave technique for extracting soil water. This is a centrifuge technique. There's the bag technique that's become popular. This was initially done by Len Wassenaar here at Environment Canada. Him and Jim Hendry and many others now are using this technique. It's a bag equilibration approach. And then there's hydraulic squeezing, like oceanographers might use. And what we find when we compare each of these techniques, again, posing the null hypothesis that regardless of the technique, we should get back the same standard, what we find is the following. Here again is our plot of O18 versus deuterium. The red is our reference water that we're trying to get back to. Each of the samples are handled in the same way, albeit with different techniques. So we're applying these 6 different techniques here. And these are too small to read in the legend in the upper left-hand corner. But what you're finding is quite a bit of spread depending on technique. And you can see some of the variations in terms of per mil values for O18 and deuterium. If I go back to these Z-scores that I wanted to show you earlier, this helps to quantify those differences. So here's the microwave extraction, the vapor equilibration in the upper right-hand corner, squeezing, cryogenic extraction, and centrifugation. And what's interesting with these results is that these Z-scores, that's just a ratio of the extracted minus the reference water divided by the standard deviation. If you look closely in that kind of a bullseye there that you see, it's suggesting that centrifugation and squeezing are 2 of the better techniques of the ones that we've tested in terms of the ability to get back the reference water that's been added. Okay, so let me try to finish off then. Maybe make one comment before I finish off, and that is if you paid attention to the first part of the talk, you may be having some ecohydrological separation anxiety or thinking I'm talking out of both sides of my mouth here. I think the point I want to make before I move on is that while this is problematic for soil, building on what others have found, We've not seen these effects with plant water, although we're actively conducting extractions and intercomparison of methods with plant water this summer. And soil water still shows a strong evaporation line regardless of method, although there's wobble depending on what method you're using. We're now working with field soils rather than lab soils or soils that are purely a construct in the lab to do these intercomparisons also this summer. So I hope to be able to report on that soon. The last thing I'd like to touch on are some very recent targeted experiments to try to go after some outstanding questions. And again, there's just so many questions and so much in the way of critical analysis that's still warranted with this whole idea of ecohydrological separation. We've gravitated to the tropical rainforest biome at Biosphere 2. This is a site that is a tropical biome. You see some of the pictures here. You think you were standing in an Amazon rainforest. There are towers inside. You see the biome in the upper right-hand corner photograph. There's 23 main tree species, 7 dominant canopy species. It's about 1,500 millimeters of precipitation a year, and that's applied weekly with 3 10-millimeter events. So it's a wet place. And what we've done and what I'll report on is a 10-week drought experiment to go after some questions. And critically important, based on what I just said, these are gravelly textured soils. So here, more than anywhere we've worked, extraction is infinitely easier. We're going to be showing you cryogenic extractions, but as I showed you with the sand, these gravelly soils don't suffer many of the issues that I've been talking about. So a little bit on the drought. On the x-axis is time. The shaded bit is the 10-week drought, and on the y-axis are a few different factors. You're seeing transpiration rate in the green in the middle, decreasing through that drought as expected. You're seeing vapor pressure deficit on the upper 2 bars, and you're seeing soil moisture in that lower bar. And you're seeing the blue and red and dashed lines showing that as we initiate drought, we have declining moisture content, and then after we finish the drought and resume precipitation, we get a rebound. Now this is the point I want to talk about, because when we resumed precipitation after the 10-week drought, after we stressed the plants to ask where are they getting their water, we added a label to that rainfall to be able to trace that water and its extraction from the ecosystem. So what I'm going to show you now on the y-axis in this large plot on the right is the deuterium composition. We added a very high deuterium label, and so the label is responding to this plus 200, I think it was, per mil deuterium label. And what you're seeing is the xylem water response to that after the tracer injection in the vertical dashed line. And what you're going to see, this is for one particular species, C. pentandra. C. pentandra now looks quite different to C. racemosa and some of the other species in terms of the uptake of this tracer that we've added at the cessation of a 10-week drought. And the C. pentandra is a drought-tolerant species. The racemosa is a drought-sensitive species. So already you're seeing some differences in terms of whether they're drought-tolerant or drought-sensitive. The uptake of water and tracer is quite different. We can quantify as well the age, or the transit time, I should say, of water as expressed by the xylem water that we're extracting. So here is just a visual of our ability to sample through the subsurface. We also sample what comes out the base of this critical zone. It varies from 1 to 3 meters, and we have ways to collect the seepage. This might be equivalent to groundwater recharge. What you're seeing here is concentration on the y-axis versus time, and this is the breakthrough following the re-wetting after the drought. And the re-wetting is taking place in this period of time in the gray band, as you can see. And what we're seeing for seepage in the blue dots, a fairly rapid appearance of tracer, on average about 8 days for that peak of the tracer to show up in the seepage, and then a quick tail off. But when we contrast that with these 3 species, you saw racemosa in the earlier slide, these are 2 other species, what you're seeing is a very different breakthrough. So these are gamma distributions fitted with this transit time model, just showing you now these very different transit times for uptake through the vegetation. So this is now the exit time, if you like, of roughly 24 days, 29 days, or 63 days, as you can see for the species here in gray. Now this issue of the time it takes, or the different time scales, has been central to this thinking about kind of two water worlds or ecohydrological separation since the 2010 paper, when there were analogies used comparing the water in the tightly bound soil to what might be moving on a slow country road to the water that's moving in a mobile fashion like a superhighway. And comparing these two is what we're trying to get at here. And the first we've been able to try to do this under controlled conditions. And what we're finding is that generally at this B2 site, the leaf soil water potential gradient is what's of course driving uptake. If we look at this on the y-axis, this is the tracer velocity on the y-axis versus that leaf soil water potential difference, if you like. But what we're finding is that travel time is strongly species dependent. And you see kind of a cartoon of that on the right-hand side, particularly when you are able to contrast the drought versus post-drought, and then look at the resulting ages of the water that's making its way through the critical zone. Now the last example, very briefly, that I want to show you before I wrap up is continuing on this idea of how old is this transpired water. This is something that's very difficult in a natural setting to quantify with O18 and deuterium. So we've gone to the Loess Plateau, working with Bing C and his colleagues at Northwest Agriculture and Forestry University, and gravitating to a site that has some rather unique attributes. So you all know where the Loess Plateau is, north of Xi'an. It's an area the size of France. There's been a lot of revegetation going on in this region. And one of the things that have been planted are apple orchards. And at one of these apple orchards, Bing and his colleagues have a beautiful record. And you can see here the depths down through the Loess, 0 to 20 meters depth. They have a beautiful record of volumetric water content at different times through the year, but also the tritium composition of that soil water. So tritium is another isotope. It's radiogenic. This peak would be associated with the 1963 bomb peak. So this is looking at the pulse of water through the unsaturated zone very slowly over time. So you can see since 63 till now, roughly 53 years or so, it's taken that long to get 8 meters down through the unsaturated zone. These are very, very deep unsaturated zones with the water table down more like 30 to 50 meters deep. But taking advantage of this tritium profile, we can squeeze the apples. We can actually quantify the tritium composition of the apple. We don't have issues like isotope fractionation to deal with. We do have the half-life of tritium to deal with, but if we factor that in, what we find in terms of matching the apple water to the age of the water now down through the soil profile based on the tritium composition, these apples are about 29 years old in terms of their water age. And if we look at the distribution of the water that's being sampled in the critical zone by these apple trees, what we find is it's fairly evenly distributed. This is a Bayesian mixing analysis for 5 trees. So here the numbers on the left-hand side are the tree numbers. These are just 5 apple trees. And with this Bayesian analysis, you're seeing then these different colors pertain to different depths through that profile. So the gray is 0 to 2 meters, there's a green 2 to 4 meters, kind of a purple from 4 to 6 meters, and so on. And what you're seeing is this variation from tree to tree. So to kind of wrap up, I guess what I'm saying is on the right-hand side, there's been a lot of work in catchment hydrology and critical zone science looking at the age or transit time of water through soil profiles in streams. There's a whole cottage industry of work on transit time of stream water, relating it to groundwater age, catchment characteristics. And what I'm showing on the left-hand side is trying to get at some of these ages of tightly bound water that we've not yet looked at. And what we're finding is, interestingly, this tightly bound water, at least in this one field case, is varying from, say, 0 to 50 years, and the water that the trees are using is on average about 29 years or so. So let me try to wrap up. And I guess what I'm saying is that some of these findings, not just with our group, but I think all of these groups that are looking at stable isotopes in the subsurface are finding that these are at odds in many ways with some of our benchmark studies. This is one that I've loved and followed for many years, Horton and Hawkins 65. This was really the foundational material that Hewlett used to base the translatory flow concept on. Flow path of rain from the soil surface to the water table. And you know, what I've been saying this morning is really at odds with this view as expressed here. Just a quote from the paper, our studies show that the percolation of rainwater through the soil to the water table is accomplished throughout most of the flow path by downward displacement of water previously retained by the soil at field capacity. So what we're finding is anything but translatory piston flow. Now, interestingly, these findings in China are suggesting something of piston flow on the long time scale through the micropore space, even though there's preferential flow of the more mobile water at or above field capacity. So there is so much to learn, I think, in this regard, and going after places to perhaps yield information, whether it's controlled experiments or places in the world where we can capitalize on certain boundary conditions, I think is a useful way to go. So to conclude, I guess I'm saying that the subsurface is acutely non-well mixed. And I think there's some support for this ecohydrological separation hypothesis, but again, I'll be the first one to reject it as we bring in or find additional data. And I think that the vegetation water use of older water is interesting, and maybe it's consistent with this differential response that we've been seeing at the Biosphere 2 site. But I think what it's all saying, all coming down to, is extreme compartmentalization in the subsurface. And I guess coming up with new methods to be able to query that water and not have those queries be method dependent is really a holy grail for us in critical zone science. The last thing I'll throw out there, and this is where we're headed, is to try to incorporate some of these findings in our models. And there was a very nice paper by Marcus Ratchevich at TU Delft that came out earlier this year. It's an overview paper in Wires Water talking about transit times and how this can be the link between hydrology and water quality at the catchment scale. And much of this paper kind of lays out a vision for how we begin to bring in some of these findings that the isotopes are providing to ultimately come up with models that are giving us the right answers for the right process reasons. Thank you very much.